Okay, so as I'm recording this, this just came out yesterday, and it's this preliminary findings by Pfizer about their vaccine here, which is called BNT162b2, which is the mRNA vaccine used against COVID-19 or SARS-CoV-2. And they were looking at whether it still works against this new mutant that was first found in England and is now spreading further. It's been found in a few places in the U.S., but that's this asparagine 501 to tyrosine mutation here, which I've talked about in previous videos. And so I wanted to go over these findings a little bit. So this is the one figure that they actually have in this short paper. And they did a plaque reduction neutralization test, which I have outlined a little bit over here. And so what that is you take these vero cells, so in this case they would be cells, for instance, that maybe like lung cells that contained the ACE2 receptor that the COVID-19 uses to actually get into the human cells. And you would grow these up on a plate like this, and then the next day you would add, so this, I got this from something that was looking at the Zika virus. That's where this particular figure came from. But you'd have the Zika virus and serum, which is essentially sample that you pull from somebody that contains the antibodies that you are interested in. And so in this case it would be antibodies against the spike protein of SARS-CoV-2. So I'll cross this out here and put SARS-CoV-2. And so then well, I guess the first thing I should have said is you actually use different concentrations of this. So you want to do dilutions of it, and that's what this is showing, where on the left side this is the most highly concentrated, and then you do what's called a serial dilution down the line, so that once you get to this end it's the least concentrated. So you're testing at different concentrations of this antibody here. And then you want to acquire the data here. So this shows that they're using luciferase substrates. There are multiple different ways you could get the data from it. But essentially what you're looking for is how much these cells were infected. So you're looking for how infected the cells were, and sometimes that's called plaques. And so that's why it's called a plaque reduction neutralization test. And then you want to take all your data and plot it. And so what we see here, this is the infection rate essentially. So when we were at our highest concentration of the serum, we had very low infection rate, and when we had very low concentration, we had high infection rate. And what they're looking for is, this called it the NT90, but in this they were actually looking for the NT50. So they were essentially looking for, well, what they were doing is taking this data and they were comparing it to cells where there was not any serum, and saying at what point did we get 50% less infection, so that'd be somewhere like around here, compared to the virus that did not have any of the antibodies added. And so that's what they are showing here. So I found this kind of confusing. I'm guessing this is the amount of dilution up here. So I would think that this would be the low concentration, and this would be the high concentration. And so they were looking at this is the wild type, and this is the new strain right here. And so what they were looking at here was the different serum samples that they pulled from people who had been inoculated, who had gotten the vaccine. And I think the triangles represent people who got the where they took the sample from them 2 weeks after they received their second dose of the vaccine, and the circles are the people who received or who had their samples taken 4 weeks after receiving their second dose of the vaccine. And essentially, what they seem to find, well, I think they have it right in the paper here, um, where it says, yeah, the ratio of the 50% neutralization of the sera against the Y501, which is the new strain, the mutant strain, to that against the N501, which is the quote unquote normal strain, was 1.46, indicating no reduction in neutralization activity against the virus bearing the new strain, the Y501 spike protein. And essentially, what they're saying is that they took the 50% for the wild type and the 50% for the new strain, and it says here the ratio of neutralization against the new strain to the old strain. So they took the 50% or the NT50 for the new and divided that by the NT50 for the old, and that was equal to 1.46, which they apparently think, well, they probably have done statistics for it, but that is not a statistically significant difference. And the one thing to note is that with these NT50 type tests, the higher the number, the equals lower neutralization. I'll just put it like that. And if it's lower, then it equals higher neutralization. And because this is looking at the concentration at which neutralization happens. So if it has to happen at if you need a higher concentration of the antibody, then you are not neutralizing as well because you needed more of it to do the same amount of neutralizing. And so if what we see here is that the NT for the new was somewhat higher than the NT for the old because our ratio is 1.46, which is greater than 1, and it would be 1 if these two things were equal to each other. And so there is some slightly higher NT50 for the new, but like I said, they have found that this is not statistically significant. However, and they point this out in the paper, a limitation of this finding is that the Y501 virus does not include the full set of spike mutations found in the rapidly spreading strains in the UK and South Africa. But they say that nevertheless that the preserved neutralization of the Y501 virus by their vaccine elicited human is consistent with preserved neutralization of a panel of 15 pseudoviruses bearing spikes with other mutations found circulating in the SARS-CoV-2 strain. So essentially what they're saying is, and I went over this in an earlier video, is that the new strain, so it's this N501 mutation in the spike protein that is sort of the one that's of most interest because this happens where the spike actually binds to the ACE2, so the human ACE2 protein. But there are other mutations that are found in this new version. I believe in the spike protein there is also a deletion. I talked about this in my previous video. I don't remember offhand where this deletion was, but I believe it was a deletion of 3 amino acids. and that was very near the cleavage site, where the S1 and S2 are cleaved at the furin cleavage site. And it seemed like there was one other mutation. I don't remember exactly offhand, but you can check out my previous video where I talk a bit more about that. But anyway, what they're saying is they have looked at this in isolation, and that's what this data here is from. And they have also looked at their vaccine against some of these other mutations. However, they have not looked at whether this data works with all of the mutations at the same time. And so that is sort of a grain of salt with which you ought to take the data from this. Um, but but from what they are saying, it seems that the Pfizer vaccine should still work against this new strain here. Um, and so I'm not going to make a call on that one way or the other. I don't know enough information about it. I would imagine if it's working against this, that it's probably going to work. But like I said, I'm you know I'm a scientist, but I'm not. This isn't my area of study, and I am not a medical doctor, so I can't really make that call one way or the other. But I just thought, given that this is the newest data on this situation, that uh, you might be interested in learning what people were saying about this. And so anyway, I hope this was somewhat illuminating for you, and I will see you in another video.